I used to record on the cloud. That's what I was talking to them about, the IT, but it's just going to go on this computer. But yeah, when we guys are done, I'll send it to you. So yeah. Okay. Other than that, okay, fair enough. So, anyone have any ideas about the first question? Who that might be? Robert Koch. Okay. So what was? Can anyone name what Koch was famous for? What process he sort of thought about for microorganisms? Postulates, right? Yeah. So really, the postulates were kind of saying, we need to kind of discover, can a bacteria actually cause a disease, or is the disease linked to this bacteria, right, or some microorganism? And so really, the Koch postulates were essentially a series of steps that basically allowed us to do that, right? I'm not going to go over that because we don't have to, but so basically Robert Koch, I think he was more famous for the Koch postulates, right? So I wouldn't say it's actually that for this one. Well, okay, any other clues as to who may or may not be? What's that? Yeah, I'm gonna go ahead and say that's the right answer. Yeah, and kind of split that a little bit. So yeah. Right. So check it out. Early 1870s. Right. Oh yeah. Sorry, guys. I forgot to. Okay, can you guys in Zoom see that now?
Oh, you're talking about the quiz for last?
Yeah, we can see it now. We can see it.
And yeah, there's some guy here. I got to. Oh, okay. All right. Anyway, so yeah, it says so basically in the early 1870s, you have the German botanist Ferdinand Kohn, and that's exactly what he did. He wrote several papers on classification, right? And it's interesting because when you study these things, they have all kinds of shapes, right? Some of them are spherical, some of them are kind of like rod looking, right? Some of them are long rods or short rods, right? And so, he was kind of thinking, maybe we can classify these bacteria according to shape, right? We can group the sphere looking ones or the cube or not cube, circular or whatever, right? And yeah, and so basically that's what he tried to do. He looked at all kinds of microorganisms regarding shape, and that was pretty much in the 1870s. And he was German and a botanist. That just means he looks at plants, right? So yeah, that's the first answer for that one is Ferdinand Kohn, right? Okay, so for this one, I'll give you guys a few minutes to think about this one. For those of you who just came in, you can use your notes or whatever. I just encourage you to think about trying it without your notes, right? Okay. And so yeah, for you guys in the Zoom, I encourage you to do the same thing. Try to think about this. Okay. So I'll give you like another minute here. Okay, I think we're good. So anyone have any ideas as to whom this can be and what they studied? So one thing's for sure, do you think it was probably the same guy who was just looking at shapes? No. Probably not, right? No. Okay. So what else? Who else do you think? Guesses? Did you say B? No. B? Okay. Well, let's check it out. So in 1970, this guy pointed out, okay, excuse me. This was the late 1970s. Yeah, so there you go. So he and his colleagues studied the fact that, or not really a fact, but just so much that he determined the nucleotide sequence of the ribosomal RNA, right? And see, that's actually pretty important, right? Because, you know, the RNA is a relatively, and especially for the ribosome, right? Those are the things that make proteins for the cell, right? And so really the RNA is kind of important to study because it's actually very stable, right? And so what we can do is we can do studies like genomics and all that stuff where we can make databases of this very stable RNA. And so then we can, you know, look at bacteria samples in the lab and we can compare the RNA samples, right? Okay, so that's why it was so important to actually determine the nucleotide sequence of this. Okay. And yes, then he did say, well, based on the RNA or ribosomal RNA rather, you can classify prokaryotes into two major groups, which is the archaea and bacteria. Okay. Alrighty. So yeah, that would be B. Okay. Go ahead with this one. Yeah, I didn't say I couldn't, so yeah. Okay, it's a good observation. All right, any other observations or maybe ideas as to what these words can mean? Chronometers, right? Yeah, so chrono kind of means like, you know, when we think of chronology, it's like a length of time, right? And so that's probably a good guess, right? And honestly, I was going to talk about it, but these questions are pretty detailed. They pretty much tell you what's going on, right? And so, yeah, we have these genes inside of us, right? And, you know, we really don't want the really important ones that code for all kinds of receptors and stuff, even with us, to malfunction, right? You know, like cystic fibrosis patients, essentially they have some sort of defective transporter, right? And see, that's really bad because they get a lot of mucus and, you know, it's a pretty horrible thing, right? And so really that's kind of part of that housekeeping gene. If you get a mutation in that, it's really bad for the organism, okay? And so it even says high mutation rates in these essential genes would be lethal, right? Okay, and so really these are kind of conserved, right? So you can look at bacteria all the way back then and see that we don't really want to change these really essential genes because that can lead to defects, right? And so we're going to conserve those even between the old species and the new ones, right? And so yes, we would actually call these evolutionary chronometers or chronometers because they tell us about the evolution. And remember, evolution is when you change your gene frequencies over time, right? And the chronometer part just means it gives a length of time as to how long that might have occurred or something like that. Okay? And see, it's not really a phylogenetic tree, right? Because really the phylogenetic trees, those are the ones that kind of branch out, right? That kind of tells us like where species diverge and things like that, right? That doesn't necessarily tell us about essential genes. Okay? And we don't really call them phylogenetic genes because it's kind of ambiguous. I mean, sure, we could use genes phylogenetically to classify things, but not necessarily if they're, you know, lethal or not. Okay, let's go to this one. Let's see. Okay. Anyone starting to think about anything maybe? Maybe like the average nucleotide identity thing, right? Okay. So let's take a look, right? Okay, so average nucleotide identity. So that was mentioned about kind of a little bit down in the chapter, right? So these don't necessarily go in like super order, you know, they're kind of spread out. So. Da-da-da. Okay, getting there. Okay, so here's kind of where we're getting to things like that, restriction. No, I think I might have passed it. Yeah, sorry about that. I think I passed. Maybe I can. No, they don't have that. Okay. It's around here. I know that. Okay. Let me see here. I think they mentioned it here. They do not. Okay. Well, I could have sworn I just saw it when I was reading in the text, but so do you guys know the difference between like species and strain? What that might entail? Yeah. So the serotype. Do you mind explaining what a serotype is? If you know.
It's like classification.
Right. Yeah. So the serotype, I would say that's you know how some microorganisms have different like proteins on their surface and stuff. Yeah. So the serotype is like basically how those proteins differ between different strains, right? Okay. So this is saying, don't worry about like the whole genome thing. That's just kind of a fancy word for saying we look at the DNA, right? And then it says the average nucleotide identity is 96%. Okay. So in general, does that sound pretty related or kind of distant? Yeah, I'd say that's pretty related, right? So obviously, now first of all, this GC content thing, that really has to do with the GC part of the DNA, right? That doesn't necessarily tell us anything about average nucleotide identity, right? So that was more for hybridization, which we'll get into. So not really that one, right? And you know, may or may not be related. Well, we have 96%. That's pretty good margin, right? And so, in fact, I think I just remembered where it is now. Excuse me. It's definitely down here. Basically, we're talking about how, oh yeah, okay. So they don't really use the word here, but see, they talk about hybridization, and really that's kind of related to this, right? Because you can take like a DNA sample and then you can break it up. And so, and this is for the unknown organism, right? And so you can have the known DNA, right? And so you can see, and evidently, you know, if you have the unknown DNA and the known DNA, you can put them in like a little sample, right? And if they kind of come together because they're the same thing, right? That's how we know they're kind of similar. And so in this case, it says two bacterial strains that show at least 70% similarity are generally considered to be members of the same species, right? So this is probably telling us, hey, they're definitely related, and it's not so much the strain level, because that's what you were saying about the serotypes. So really, this is about the actual genomic species. So I would go with A. Okay. All right. Oh, sorry. A. Right. Yeah. So it looks like at least 70% similarity. So you can think of that as average 70, right? Generally considered to be members of the same species. Right. 83% for the same chapter. Okay. I mean, that's probably true. I'm just,
Yeah, when we went over it in class, it was 83% for the same genus and then 96 for the same species.
gotcha. Okay. So, well, in that case, 96 would be for same species, right?
Yeah.
Okay.
And then 83 is genus, and then 70 was just they're similar.
Sure. Okay. Good. Yeah. Thank you for clearing that up. Appreciate it. Okay. So does everyone get that now based on what he said in lecture as well? Okay. Yeah. Alrighty. So what about, let's go to number 6. Okay. Does anyone have any ideas about, so what about the first one? Sound familiar? Okay. Well, really, this one is definitely an example directly in your book. Let me see. It's gotta go up here. And so really the main point of this question is saying, well, obviously which one's true, right? But you want to think about certain things. So for instance, this one, right? The Troporema whippili. I'm not the best with the names. So really, as it turns out, a lot of microbes can't be actually cultured, right? They're just we don't have the conditions or something. In fact, only like less than 2% of ones that we can cultivate in the entire world are the ones that we can actually use on like plates, right? And so this one, so we can actually use this rDNA or ribosomal DNA to identify the uncultivatable organisms, right? And so let's just look at the DNA instead of having to grow it and then do all other tests, right? And so really this one was actually cultivated, or excuse me, it was found using DNA even before we can actually cultivate it. Okay, so that is true. Okay, this is identified before it could be grown, right? And then this one is about the GC content of DNA. So remember what the AT and GC stuff is, right? From just regular biology. Yeah, so that's kind of like the nucleotides on the DNA, right? Okay, and so the GC one, does anyone know why that one is kind of important versus the AT? Think about like bonding, by the way. Yeah, so the GC one, that's going to have like 3 hydrogen bonds, right? So if I look up bonding. So let's take a look at this. Yeah, so notice the GC has these 3 hydrogen bonds, right? You don't have to know the structure, I'm just showing you. And the AT only has 2. So this suggests that, well, a lot of the CGs, it's going to be a little bit more of a tighter bond, right? Okay, and so this question is saying it can be measured by determining the temperature at which the double-stranded DNA melts. Okay, and actually, we can take a look at this. I think it's down here. Okay, so here we go. The GC content is often measured by determining the temperature at which it denatures or melts. Okay, so that is a true statement. And they even kind of show you, saying like, here's by the way, they kind of go a little fancy with the absorbance and everything. Don't worry too much about that. They're just saying, hey, at this temperature, that's when the GC kind of breaks apart and we get the 2 fragments, right? So that part is true, B, okay? Okay, what about C? Anyone think about C? Any ideas? So what do I mean by sequence? What is that related to? What word? Do you think of anything? When you say sequence?
The DNA sequence for the
DNA
organism.
sequence. Good. Yeah, very good. So really, the whole basis of this comparison stuff is we can use DNA to look at differences between molecules, right? Because it can tell us if we're related or not. In fact, we share like 99% of DNA with like chimpanzees, right? So we're kind of related in a way, right? And so really this is saying, can we use DNA sequence differences to assess their relatedness? And I would argue, yeah, right? Because evidently, again, back to the example of chimps, we're pretty close to them in that our DNA is similar, right? So we're kind of related in a way. Okay. Now when it says relatedness, I mean, that's more about like, evolutionarily, right? Because we have the same genes and things like that. Okay. So that I think C is very definitely A and B is right? So C may be a little shifty, but we'll see. All right, and then based on DNA homology studies, members of the genus Shigella should be in the same species as the Escherichia coli, right? So let's look at that, because I remember seeing this one too. Notice a lot of things are kind of similar to your book, right? So this may also be, I'm sure this is in PowerPoint as well. Let's see here. I think I'm just gonna be faster and just type it. Okay, there we go. So let's go back. Yeah, here we go. So check this out. It says DNA hybridization studies. Remember, that's when you take DNA of the unknown and DNA of like a known thing. And they noticed that the DNA of the Shigella and the Escherichia species kind of bonded pretty well, right? So that suggests that they have the same like bases and everything. So they're kind of similar. So it even says members of the genera Shigella and Escherichia or Escherichia are quite different for biochemical tests. So that's when you do like enzyme assays and things like that, right? Maybe one can ferment sugar, the other one can't. Even though they're different for that stuff, and maybe morphologically, they should actually be grouped in the same species just because of their DNA. Okay. Right. So that suggests, yeah, D is correct. Okay. All right, and then the last one is gel electrophoresis is used to determine the serotype of an organism. Right? So actually, another part of this is, let's see, serotype. So yeah, as someone said earlier, the serotype, yeah, that's like where we have a bacteria and it's got like different proteins on it, right? And we can compare these proteins between different strains, right? Okay, so here we go. So as you see, for instance, you might have like different antigens or something, right? So that's the serotype, right? And we want to look at this, okay? And so I believe they say, so one thing is looking at, you can phage type. That means like you can put bacteriophages in the sample and see if they eat like the same things. Not eat, but like drill into them. Let's see. And then another one is that, now this is the, looks like an electrophoresis, but that is for, yeah, looking at different strains, right? So, yeah, and so look, it says gel electrophoresis is used to separate the fragments so they can be observed, right? And so, by the way, this RFLP is what we call restriction fragment length polymorphism, right? So really what that means is we can take like bacteria and they have like circular chromosomes, right? Often. And so they have little points at which when you put an enzyme in there, they can break at a certain point. And so it can break differently for different bacteria, right? Now notice they say molecular typing, that's kind of for the gel electrophoresis, right? So I don't think it's appropriate to use gel electrophoresis for the serotyping, right? Notice it says serotyping is for like something else, right? Molecular typing is more for the gel electrophoresis stuff, okay? So it kind of makes sense that, at least I would say that it looks like E probably isn't the most appropriate choice. So I'd say E. Okay. right. Let's take a look at this one. Sorry. Okay, does anyone have any ideas on what this may be? Biotype or serotype? And this is again, if you had the PowerPoint up, I believe they mentioned this thing, for instance, right? Okay, so here we go. So a biotype is, they call it like a group of these organisms with a characteristic pattern. Okay, and so really, it's not very specific in here. So let's go back. Let's go and actually look at it, right? So, okay, here we go. Here we go. Okay, so notice. So let's think about the difference between. So notice one is grouped under biochemical typing, and typing really just means we're looking at the different. We're characterizing these strain differences, right? Strains meaning they have different proteins on the surface or interact in a different way or something, right? Okay. And so really for the biochemical stuff, it's saying, we can use these biochemical tests. Maybe we can give them a sugar and they'll reduce it or something to distinguish between these strains, right? And so notice it says a group of those that have the biochemical patterns is the biotype. Like you see that kind of wording, right? The bio from biochemistry and type meaning like typing, seeing what they are, right? Okay. And then the serological is, yeah, that's when we have the different little flagella on the bacteria, right? Or some maybe some membrane proteins or something like that, right? Or the LPS, remember the lipopolysaccharide in some of the bacteria have, okay? So in that event, the biotype is really for the biochemical stuff, right? And so let's see here. It says, so the first one says biotype is the living type of the microorganism identified, and the serotype is type of blood serum. Well, do bacteria have blood? No, right? So let's look at B. So a biotype is a biological environment, and the serotype is the blood serum. I think we just established that there's no blood in these bacteria, right? Okay, C. So we got it's the particular phenotypic traits and the characteristic structures. In the serotype, they're saying is a list of molecules it's able to make and secrete. What do you think about that one? Saw some heads shaking. Is that a no? No. Okay, yeah, because honestly, remember the serotype was saying like we can look at the structures, right? Yeah, the lipopolysaccharide or whatever, right? So yeah, I would say it's probably not C, right? Because evidently the biological structures, the biochemistry, that's more for like what it can make and secrete, right? All right, and then yeah, I think this is the best one, right? Yeah, because biotype again, like we said, bio and then biochemical, right? And then for the serotype, we're curious about what structures or antigens are, right? And those are some big pathogenicity factors. Some bacteria infect you differently because maybe they have something that's better at latching onto your cells or something, right? For instance, COVID-19, right? We have the Delta variant and the other variants, right? The Delta variant is shown to bind tighter to one of our ACE2 receptors, right? And so that tells us, hey, the serotype of that one has different proteins, or maybe it's got a better protein, right? So that's what the serotype is, okay? All right. So yeah, that was D. Okay. Oh, and yes, and it does say, it's characterized by the different antibodies in the individual serum. Well, we do have to make, when we get an infection, our body says, well, we want to make something that's designed to wipe out the infection specifically, right? And so our body goes to work saying, let's make antibodies, which will destroy these bacteria or whatever you have very specifically, right? And so that's why we can call it serum or serotype, right? All right, what about 8? Anyone have any ideas or thoughts? Maybe doesn't have to be the answer.
Could it be E?
Or how did his
E?
captain go?
Yeah.
Okay, that's
Something
definitely a
Celtic
possible
like.
choice. So why would you think that?
I don't remember.
Just because I don't remember learning about it.
Right. Yeah, it's most of these are kind of like all the other ones are bogus, right? Well, we can walk through it real quick, right? So peptidoglycan. When you guys learned about that, what's so special about peptidoglycan? What about it? Gram stain, right? Yeah. So can we gram stain human cells or eukaryotic cells? No, why not? Yeah, we don't have peptidoglycan, right? So this is saying peptidoglycan cell walls can be found on bacteria. And I'd say that's true, right? Because bacteria are the ones that can have the peptidoglycan. Okay, introns can be found in the eukaryote. What are introns? Right, exactly. They kind of, that's used in DNA regulation. Specifically, let's get a picture. Really, it's part of introns and exons, okay? So here's kind of so this is kind of the DNA in all, like let's say our cells, eukaryotic cells, right? So really, what happens is, we have exons and introns. And so really, we have this whole sort of sequence. And what happens is when we make it into the mRNA, remember the dogma, right? We go from DNA to RNA, right? So before we can make the protein, we need the RNA. And so as you can tell, notice how some things are just straight up taken out, right? And so we call those introns. And I know it makes no sense. It's like in, but it's out. Sorry, I'm not the one who makes this stuff. But let's just say that, as it turns out, bacteria do not actually have that. Okay. Bacteria just kind of have one long thing going for itself. Okay, we have the introns and exons. So absolutely, we can find it in eukaryotic cells. Okay. All right. 70S ribosomes. That one's a little specific. And really, this whole question was definitely one of those, like, let me see here. I remember they set it up here earlier. Okay. No. Aha. Okay. So check it out. Presence of introns. Eukarya has it. Bacteria don't. Sometimes archaea. But really, we're curious about the ribosome, right? So as you can see, 70S ribosomes can be found in archaea. Is that true? Yes. Okay. Membrane-bound organelles can be found in the eukarya. Yeah. So what are those anyway? Like, you guys know what those are just in general? Organelles. Have you heard of mitochondria? Right. Powerhouse of the cell. Yeah. So we have, Golgi apparatus. You name it. Those are the membrane-bound organelles. Absolutely, eukaryotes do have that. Okay. And then this one, E, obviously was the one that somebody said before, kind of like process of elimination, but it is true. We do not, excuse me, there is not, excuse me, there's hydrocarbons, but not fatty acids in the archaea. Okay, so that is E, not correct. All right, how about this? Okay, so what's McConkey agar? What is that? What do you mean by plating it onto some agar? What does that mean? Any ideas? Doesn't have to be right. It can just be whatever you think it is. So what about in your labs, in your actual micro labs? You know those petri dishes, right? So we can take. So those are very simple ones, right? But the idea here is someone has a UTI, maybe. We can take their urine and do some things with it so we can get parts of it we don't want, and we can pretty much place it onto this McConkey agar, which is like an actual media plate, right, an agar. And we want to see if it, and McConkey agar is special because it has like lactose in it, right? And so if the bacteria can ferment lactose, the lactose will make a product, or rather the bacteria will make a product of it, and that product will turn pink on the agar, okay? And really the question is it true or false? Will we get a pink agar for E. coli or not? And I believe the answer is actually true. We should expect to see a pink agar for the E. coli. And to confirm that, yeah, that looks pink to me. All right, it looks good, but don't eat it. Alrighty, so yes, that was yes for that. True. Okay. What about this? This one's kind of like a puzzle. This is like the real micro questions, right? What is the bacteria that is hurting or causing an ailment? Right? Okay, this one is a little bit difficult. I do encourage you to think about several things, honestly. Okay. So this is kind of part of the phylogeny chapter, although really, to be fair, this is not really phylogeny, right? Because we're only talking about certain aspects of the bacteria, whether it's got peptidoglycan and it's gram positive. Although, okay, you can have peptidoglycan and gram negative. I mean, the way it's arranged, right? Okay. If it's gram positive, it happens to have an enzyme catalase. Anyone know what catalase does, by the way? Yeah, it does form bubbles, probably because of a reason. And I believe that reason is because it can take hydrogen peroxide, right? And then we can, and really, you got to think about the reason why that might be the case. Well, you know, our cells too, when we have oxygen, right, we're going to use it. As it turns out, sometimes we can make reactive oxygen species, which is really like, you can think of it as like an oxygen, and instead of having like the normal electron, sometimes it has like an extra electron that's unpaired. It's going to want to, and this is caused by metabolism. So bacteria that use oxygen will probably have catalase. And the reason why is because when we form this, they want to have a mechanism that they develop to get rid of any dangerous things, right? H2O2, hydrogen peroxide, is definitely one of those bad ones, okay? So catalase serves as to get rid of this. So really only bacteria that use oxygen would have something like this. That's kind of a clue. Coagulase, okay? That one's, what does coagulate mean just in general? Like when you think of coagulate. Yeah, clog, like clog, right, exactly. So really, if you have coagulase, you can turn, like in the case of our cells, we have fibrin, right? Or I believe it's fibrinogen, something like that. And we can turn it into, you know how the clot kind of is, right? You got a bunch of the blood cells, right? And then you have this weird stringy stuff all over it, you know? Okay, well, really, this is like the fibrin, I believe. And so some bacteria can straight up make you have clots, and it could be all over your body. It could be systemic. Okay, so it's coagulase positive. Okay? And then we have some options. Okay, not the easiest things, right? So honestly, this is probably the best to look at kind of an earlier thing they had here, based on some chemical tests. So I saw it. Okay, this is probably what we're curious about. Okay, so let's take a look. So it turns out we can gram stain it, right? And so it is gram positive, right? Or sorry. Okay, we are gram positive. So that means we have a coccus. Okay, so pretty much, well, that tells us, hey, it very well could be this, right? Because it's gram positive, right? Okay, that's one thing. Then catalase, okay? If we have the catalase enzyme that can get rid of oxygen, it means it's an oxygen using bacteria. If it's positive, which I believe, yes, this was catalase positive, it can be determined if it has coagulase or not, okay? So it's kind of an intermediate thing. You can't go directly to something. So it is gram positive, it's some coccus, and it is catalase positive. So that means it depends on coagulase for the deciding factor, right? So as it turns out, if we're positive for coagulase, we'll have something like Staphylococcus aureus. And if it's coagulase negative, we might have something like this saprophyticus or something, okay? So it is coagulase positive. So what does that tell us? It probably is Staphylococcus aureus, right? Yeah. And that one is kind of bad. That can cause quite a few infections. And here's kind of where this is going to come in, just to show you guys that this stuff is real. Okay, so check it out. In species, we have this Staphylococcus aureus, and check it out. Look at that enzyme coagulase. Okay, and that's why it's a useful diagnostic test. Okay, and it can cause disseminated like blood clots over your body if you get infected by it. So don't. Alrighty. Just kidding. Yeah.
So is it? That's what it shows that.
Right. Right. It's just in this chapter, they kind of showed you an example, right? So yeah. Because honestly, like even this is too little. There's probably hundreds of thousands of millions more bacteria that could be like this. This was just part of this context. Okay. So yeah, we said it was Staphylococcus aureus. All right. What about this? Diazotroph. Yeah, this is probably getting into the other chapter. So let me go ahead and 11, right? Okay. Okay, sorry about that. Alrighty, so one thing's for sure is it can fix atmospheric N2. What does fix mean just in general? Does anyone, and I don't mean like you fix your broken bike. I mean like in this context, do you know what fixing something might mean? Conversion, right? So what are we doing about that? So it's saying we fix specifically atmospheric N2, right? Okay, so can we as humans, can we use nitrogen? Like, I mean, we breathe it, right? But most of the oxygen we use, then we breathe out the nitrogen. Can we use that nitrogen as like reactions in our cells or something? See, I'd say no, right? Because it's just a gas, right? We can't do anything with that. So these bacteria fix the N2, meaning they can turn it into a more biologically available form, meaning we can use it, or we can use it as fertilizer or something, okay? And so that's what the fix means, okay? Anyway, and then kind of the actual question is, which of the following genera is a diazotroph? Okay. And then it says, here's a hint, species of this genus can be polyploid under some growth conditions, and it can have 80 copies of its chromosome. Okay, so that's what polyploid means. You can have many copies, right? So honestly, this is probably one of the more easier look in the PowerPoint questions, right? So, so let's take a look. Okay, so this was about the archaea that can, okay, diazotrophs, right? So let's see. Okay, never mind. How about this time? Okay, see what we get. Okay, here we go. All right, so here's an example. Among the free living examples, we can have Azotobacter, right? But we're more curious on, oh, well, it says genera, right? So That is a genus. That means genera, same thing, genus, right? So honestly, yeah, that's probably our answer, right? Because it gives it to you. And these other ones, I mean, we could go one by one, but kind of running low on time. So I think it's better to just realize that, notice that was in the under diazotrope frame. Okay. As far as the polyploid, that one's a little interesting. I'm not really sure about that one. It may say that here, I'm sure. Maybe it even says it in the PowerPoint. Let's see. Oop, yeah, that one was a little specific to the book. So, all right, no problem. All right, genera most likely find growing in acidic runoff from a coal mine. Does it matter that we have a coal mine? Like, is there a bacteria that wants the coal or something? No. Just pay attention to where they say acidic runoff, right? So that means bacteria that can likely survive in an acidic environment or a low pH. Okay, so that may tell us something, right? Also notice thermos, right? What do you think when you usually think of like thermos or thermo? What does that really mean typically? Like heat, right? Yeah, so that's probably something you'd find in those weird underwater, those freaking volcanoes or vents they have. Those are hot, right? That's probably the thermos. Okay. And yeah, so you did say, well, maybe the one with acid in it. Well, there's two, right? So there's lactic acid and acidithiobacillus, right? So yeah, this one's kind of just looking at the text and kind of compare, right? It's likely, yeah, I believe it's this one actually, but let's see here. No, he didn't. Anything it's from you?
I saw it when I was
Okay, yeah. And I'll ask for the answers to send to you, by the way. I'm not just gonna leave you guys hanging. And I'm not just guessing on these, like I actually looked at this before. So okay. All right. So what about this one? Environmental extremes. So extreme temperature, extreme acid environment, whatever. Okay, so remember this one was kind of like, well, that was for like the nitrogen, right? So that didn't really tell us about any extremes environmentally, right? Okay, what about Clostridium? Do you know, what about, is there something special about Clostridium and Bacillus species? Endospores, right? Is that a useful evolutionary thing? Right, yeah, it is, because we can definitely hide from some, maybe acid in somebody's stomach or something, or we can, some disinfectants we can avoid, right? So that's probably something as well. We do have this myxobacteria. Now that streptomyces, that seems more like the more common. I don't think that would be associated with, see, most of these names kind of remind you of something. Strepto, that probably just tells us it's not necessarily like spore forming or something, right? So yeah, that Clostridium and Bacillus is looking pretty good, right? So that being said, I think there was something about that in here. And I'd say, I mean, yeah, here we go. So yeah, so check that out, Bacillus, Clostridium. So it looks like he does list Bacillus first, right? And this was 11.6, right? So let me check. Oh wait, that's something else. Sorry. It was E. Okay, thank you. And that kind of, it definitely does have acid in the name. So yeah. Yeah, so for this one, yeah, it looks like probably we're comparing Bacillus and Clostridium, right? Because those can form the endospore, and the endospore is really hard to get rid to kill, right? And so really, we're going to be looking at probably Bacillus and Clostridium. And as far as most resistant, so it's not, we don't really care about 1, 5, or 5, right? So probably 2 and 3, right? Okay, not necessarily in that order, just 2 and 3. Those endospores are really tough. Okay. All right, this one, PCR. think that was chapter, if I'm not mistaken. So that PCR wasn't really exclusively in the chapter 10 where they talked about the techniques like that. I think that was chapter 9, if anything. So yeah, this might be kind of a review question. Okay, yeah. Well, in that case, here we go. So. Oh, there you go. Okay. B. Okay. Yeah. What do you guys think? So we can just, kind of look at the steps real quick. It's a very known thing, and it's not super hard. Okay, so we can have this. There's so many good pictures online as well. These are just a couple. So yeah, let's look at this one. That's a little small. Sorry. All right, let's just look at this one. So yeah, we can take the DNA. Now, obviously, we have the DNA, right? And so in order to actually look at it, we kind of have to break apart or denature it, right? The bonds. Bless you. And that serves the purpose of why is it bad when we like. So that's one reason. So why is it bad when we get a fever? That's okay. That's more about proteins, actually. I probably shouldn't say that. Okay, how about like radiation? So why is it bad to get large amounts of radiation for our DNA? There's a lot of reasons, but how do you think this relates to denature? Right, exactly. So the DNA, we kind of need it to be together so that we can make a mRNA out of it, right? And so if it's denatured, we can't do that, right? And so what this does though is we can say, well, you know what? Let's use PCR. It's like what our body does, just more fast, basically. So we take our double-stranded DNA, we can heat it up really hot and denature it and take it apart, right? And then we could start what we call annealing it. So remember, when you have the DNA, so here's our DNA. Okay, so imagine we break it up, right? So now we got different things. Okay, so now, we'll separate, this will like float off in solution or something, right? Okay, so what we can do is we want to make a copy of this, right? And so what we can do is, remember, you can't just start adding nucleotides onto this thing, right? And remember, these little things are nucleotides. You're actually going to have to prime it, right? Because polymerase, it has to see that there's something it can start going on, right? And so you need a primer, right? So you need something initially just to get polymerase to latch on to, okay? And so basically that's going to come in, do its thing, right? Although it's Taq polymerase in the PCR, but anyway, then you can actually start making it, right? So we have to denature it first, okay? And then we can start annealing by annealing these primers to it just to get something started. And then we actually start extending it. Okay? So yeah, so we got to denature it and then anneal and then elongate. Okay, so did you guys say B? I can't recall. Okay, yeah, B. Okay, that one's kind of simple. All right, DNA synthesis. Which of the following is not a key ingredient to the PCR process? Okay, so remember what I just said about, I actually just said it, Taq polymerase, right? So really, remember those hydrothermal bacteria we were talking about? So, our polymerase it doesn't operate under very hot conditions, if you will. So some bacteria do, and so we can isolate their polymerase, their Taq polymerase, and use it in the PCR because it works with heat. It can do that, okay? And so that's a very vital ingredient, okay? All right, and then pH buffered solution. So really, remember, does everyone kind of remember what a buffer is just in general? Yeah, okay, yeah, so buffers really like, it keeps it so we don't have a lot of acid or a lot of base, keeps it kind of neutral. So yeah, you want a buffered solution because if something's too acidic or basic, that can start messing up the DNA, cause reactions or all kinds of things. Okay. Let's see. So yeah, I agree with C. Oh yeah, double-stranded DNA. So what do you think about that? well, we need to get something going, right? So that's kind of the main ingredient we put in there. So I would say, yeah, we need some DNA to get started, right? Okay, and maybe I got okay. Maybe I got a little too. I mean, when we did PCR, I think we used a buffer, but maybe it's not the most important thing. I think he just cares about the actual ingredients more than the chemical stuff, you know. So, because evidently we do need DNA to start, right? And remember, we do need Taq polymerase and the primers to actually start, right? So yeah, we do need primers and double-strand DNA and Taq polymerase. So, by the way, once we got Taq polymerase on here, how are we going to start adding nucleotides? They have to like come from the solution, right? So DNA polymerase has to, there's a bunch of nucleotides that are going to start falling into place. He'll take them and he'll lay them down, right? And so really you do need nucleotides, right? To actually, they're not just going to come out of thin air, right? So really, yeah, I guess maybe the pH buffered thing doesn't matter that much. It's B. Oh, does that say RNA? Oh, excuse me. Okay. Thought I saw something else. Okay. Yeah. We do need primers though. So was he saying it's not RNA specifically or something like that? Okay. True. Okay. Yeah. Yeah, those primers are technically okay. I see what he means. Yeah. So if you were to make the RNA, you would need an RNA primer. But to make the DNA, we need the DNA primer, right? Okay. So it's just a matter of semantics, really.
He said it was also, he said specific that it wouldn't survive temperature cycling.
Oh, okay. Thank you. So, so guys on Zoom, I was told that first of all the answer is B, and that's because the RNA primers basically wouldn't survive the high temperatures of the PCR, and it's really about DNA primers. So you guys get that on Zoom. You guys good? You guys did? I really hope the mic is on. Yeah, it's on. Okay, yeah, nice. Okay. Alrighty. Alrighty. So PCR was revolutionary, of course. And oh yeah, okay. So it's just asking for the creator, right? So who do you guys think it is? What's that? Yeah, it's Mulus. And you know, if you want a way to remember this, he actually thought about this while he was doing LSD. I'm not even kidding. Yeah, he was the one who invented PCR, and he got the idea of on an acid trip, basically.
Oh, he didn't. Okay.
Oh, he invented LSD. No, no, he didn't invent the LSD. He simply used it to think of PCR. But don't think that was, don't think that's good though, because like I think he went a little nutty afterwards. But yeah, that's what things happen. But yeah, I think. Well, yeah. Okay. Yeah. Yeah. So it is true. He did use LSD. LSD dropping, climate change denying, astrology believing, crazy guy. Right, yeah. So you will not forget this guy, right? Okay. Now let's look at gel electrophoresis, right? Forget about that one for a little bit. What's that? Oh. What do you mean? Oh. For number 5? Oh, 6. Yeah, so was it 6? I thought it was something else. Right. Maybe, yeah, that's a good point. Okay, so the question was, so he was making a good relation. So the question was, um, well, really, it's a matter of just looking at it. So
That's very specific.
it is very specific, I agree, but it's a good thing to know. So sure. So yeah, the premise is kind of simple. I'll show you. All right.
Okay, this is would be positive.
Yes, and let's think about that. Why it is. So here's kind of the setup. Okay, so we got our electrodes, right? And really, those are just things that apply negative current or electrons, right? And so you can have it negatively charged or positively charged, right? So really, it's going to go this way because what charge do you think DNA has? Because it's got a net charge biologically. So if it's attracted toward the positive end and repelled from the negative end, what charge do you think DNA has? Negative, right? Good. Positive and minus don't attract, and the same charges repel, right? So yeah, good. It's got a negative charge DNA. So that means it is going to go from the positive, or wait a minute, excuse me, it's the opposite, isn't it? Because this, yeah, this one says it goes from the positive. Yeah, that is false. Yeah, we're supposed to go from negative to positive. I mean, how could we start a positive if DNA is negative anyway? Correct. But the thing is, we have to start at the beginning, right? And then we apply the current, and then it starts, right? Yeah. So we can't start at the positive because we have this stuff. Because think of the actual gel in there. Think of it like a sieve, like so that kind of makes sense, like a strainer, if you will. And so you have to the DNA has to move past it and everything and go around. If it's really big, it's gonna kind of get stuck, right? So the idea behind that is it separates by size. And then as far as moving it, the actual force that does it is the negative stuff, right? Okay, and then it goes towards the positive. Okay. So yes, false. Okay. Which of the following genera are obligate intracellular parasites, or all of them except for one? Okay. Well, here's the thing. What is obligate? I was going to say, what does the whole thing mean? But let's break it down, right? So what does obligate really mean? What is obligate? If you're obligated to do something, what does that mean? You have to do it. If you're obligated to, you have a court date, you're obligated to go to court, or so go to jail, okay? You have to. Intracellular. Does anyone know what? You don't have to get into the semantics of extra or intra, but any idea of what intracellular could mean? Right, yeah, you kind of have to be in a cell, right? So you can't live on the desk or something. You got to have a cell or something, all right? And parasite, that's kind of a broad term, right? We think of worms, right, or helminths or something. Well, parasite really could be kind of a number of, you can have if you think of a thin bacteria, technically it's a parasite, I think. So it's not so much like the word parasite, it's more just it's basically saying obligate intracellular, it's got to be inside of the cell to actually reproduce and live. Okay. So really these are all, and you'll recognize a lot of these are the cause the STDs, right? I believe well, first of all, chlamydia, right? I think it's called clap, right? Sorry, I shouldn't have said that. Maybe you can remember it, right? So yeah, chlamydia, that causes that. Chlamydia is the actual parasite, right? That causes chlamydia. Okay, and then coccia, I believe the rickettsia. I believe that one causes. Let's see. Yeah, like rickets, right?
I remember seeing that on.
Yeah. It caught okay, typhus. Yeah. So remember, all these have like a, we have conditions, human conditions, right, where you have a sickness, and often it's linked to some bacteria, right? So it looks like ricket. The causative agent, we call it, or the thing that causes the disease, happens to be this bacteria, rickettsia. So typhus and spotted fever, that's rickettsia. Okay. Can't be that one. Right. So notice, seems like it's probably going to have to be in the cell to replicate, right? You know, yeah. And honestly, I mean, sure, you can have a lot of knowledge about this stuff, but it's very contextual, right? Like you're doing this class, this chapter, you're not going to have to know everything about all the bacteria, right? Evidently, it's probably some context related to the chapter, right? Okay. Sure. T, mycoplasma. Okay. Yeah, I believe mycoplasma is, so that myco, like mycobacterium, things like that, that tells us something, right? So the myco really mean, so I was trying to find the word for it. Let's just look at mycobacteria. Okay, so now mycobacterium isn't necessarily the same thing as mycoplasma. It's just the myco tells us that it's part of some, I believe, I'm trying to think of the word. I think. Okay, yeah, it's a fungus, right? So that's a little, because mycobacterium is literally a bacteria, so it's not a fungus, but it tells us that it could be related to fungus, right? So mycoplasma, I believe mycoplasma is definitely still a bacteria, right? And let's see. Oh yeah, that's it. Okay, it's got it lacks a cell wall. That's what I was trying to think of. I apologize. So yeah, mycoplasma means it doesn't have a cell wall. Okay, the viruses, how some of them are naked. It's like that, but for bacteria. So, yeah. So really, all these are really the causative agents of a lot of sexually transmitted diseases, and that's just because the bacteria have to be in there, right? So, for instance, I think this was chapter, yeah, 11, right? So, let's see here. No, I'll type. What was the word? Obligate intracellular. Yep. On. Oh my gosh. There. Okay. Well, that's one thing. That's the only thing they have. Okay. That's not really what I want. But oh, here we go. Okay. There you go. So, yeah, the Glacis cell wall, and doesn't really talk much about it though. This talks about animals or habitats for. Oh, okay, I get it. So you kind of get the wordplay. Animals are as habitats. So what's a habitat? Just where something lives, right? Or an environment in which something lives. Well, they're saying some of these are, they have to live in something, right? And so we're animals, right? And so here we go, obligate intracellular parasites right here. So notice there we go, Coxiella, Rickettsia, and all that stuff except for mycoplasma. They just lack cells. They're just not obligated intracellular ones. Okay. So the answer would be mycoplasma. Yep. Right. Corpus Christi's water boil advisor. Okay. Okay. So kind of let's read over it, right? So there's coliforms in the drinking water, and they say coliforms are rod-shaped, gram-negative, and non-spore forming bacteria. But specifically, and this is all important to identification of bacteria, right? It tells us it's rod-shaped, okay? It tells us it's gram-negative, right? And non-spore forming. Are any of these spore forming? That's specifically Clostridium, right? So that already tells you no, that's not going to be spore. It's non-spore forming. It's not going to be E. And then the rest is probably. What's that? Okay, good, very good. Yeah, coccus is that's not the rod one. That is it's like the ball one, if I'm not mistaken, right? Yeah. Okay, so that's the wrong shape. Okay, good. All right, and then we got the Bacteroides, Bifidobacterium, and Escherichia. Well, what about Escherichia coli or E. coli? You know, like inside of us, and it helps with digestion and things like that, right? Well, is that gram positive or negative? Negative, right? Okay, so that tells us it could be a choice. Okay, so yeah, really looking at C, D, and A, right? So now we can look at, looks like rod shaped, right? So that's probably one of our, no, it does produce gas too, and lactose fermentation, right? So remember what happens if we put E. coli in, was it E. coli that we put in McConkey agar? Okay, there you go. So it does ferment lactose, right? So it's non-spore forming, it ferments lactose and makes the McConkey agar pink. Is it rod-shaped though, E. coli? I believe it is as well. Yeah, this one's kind of a little bit of research, right? So let's see.
But I mean, there's no
Yeah, bacilli. So that is true. Let's see. Oh, we're on 11. Sorry. Okay. So let's take a look. Okay, here we go. Okay, now that's something else. So maybe we'll find some clues in here. So really, for when they have questions like this where they say like, which bacteria could it be? It's a good idea to write down some popular ones, you know what I mean? So maybe write down popular lactose fermenting or something like that. And again, some of these questions, we're talking about it, right? The McConkey agar told us it was lactose, right? So that's one thing. And so this gives the phylum, right? But we're really curious about the actual genera, right? So that doesn't really help that much. So let's see. Geez. Okay. And oh yeah, what was it again? It's a coliform, right? That is true. They do. Okay, here we go. So maybe this might tell us something, right? So okay, well, we know that endospore formers gave us one thing, right? But we're curious about, so there's also bifidobacterium, right? So they don't really talk about that here. So it probably tells us, it's definitely a slide. I wonder if they talk about coliform specifically. Nope. What was the one? What was one of the names? Bifidum, right? Yeah. Something like that, right? Okay. So for instance, the ones that inhabit the mucous membranes. Right, yeah, so that's gram-positive, and it's an irregular rod. So one thing's for sure is that it's rod-shaped, so that could be a choice, right? But is this one we just looked at gram-negative? No, it's positive, right? So that is no longer a choice. So it's some. Oh, it did. You lied. Oh, Bacteroides. Yeah, so that one tells us it's gram-negative in Coccobacilli. All right. So notice sometimes it's a little like a puzzle, right? Kind of have to look in the slides or something like that. But for the most part, this is all pretty grouped together in the book and your PowerPoint. So even if you don't have the book, you're fine. It's pretty similar here. Okay. This one? That could, right, yeah, so that could be, definitely could be. All right, but anyway, at any rate, yeah, this was spore forming, that's out. This we just determined to be out due to the, I think because we said it was positive, right? Good. Okay. Okay. And then I believe we said this one, no, excuse me. We also said another one of these was out, right? Yeah, the coccus, that's the wrong shape, right? Okay. So really we're left with Bacteroides and Escherichia. And the Bacteroides one, it is gram-negative, a rod. But we're probably curious now because we got rod and gram-negative. That's also Escherichia, right? So really, I think we should look at the lactose fermentation and the production of gas, right? So let's see. Yeah, so it can be both, right? So that probably tells us, well, maybe they're looking really more for the only rod shaped, right? So that kind of gives us a clue is, and also lactose fermentation is that one, right? So I'm almost sure that somewhere in here, they'll probably mention that. And let's see here, actually. Yeah, it's pH. And heat? No, acidophiles are just mostly about, they like low pH environments, right? True, that is absolutely true. Good observation. So what I'm seeing here is that, okay, that doesn't really tell us about lactose fermentation, right? Although, wait a minute, you see that? These are gram-negative rods, and then these are gram-positive. Okay, so yeah, we should probably. Well, no, I mean, yeah. So, well, obviously the question's talking about gram-negative rods, right? And so that's what these are. And so Bacteroides could be that. Now we do know Escherichia coli can, and that's part of the genus, can indeed ferment lactose, right? So that is a strong probable answer, right, for that. But we're really seeing if this Bacteroides can either do the gas production or lactose fermentation, right? So that's what I'm trying to see here. So it looks like they say it on 298. Oh, here? Oh, sorry. 6. Okay, let's see. Okay, so it could be a coccobacilli, but oh, many are killed by brief exposure to oxygen, right? So do you think that would be in just regular open air? Probably not, right? So that tells us it's probably too stringent. And it didn't really say anything about gas, did it? Yeah, I think so. Yeah, because you can use, remember like the final electron receptor in the metabolism? I think you can produce like methane or something without using oxygen, something like that. Yeah, so that might be the case. Right. Yeah. So
Like, I'm pretty sure that's what makes it hard.
that's
Because
probably
in our
true.
intestines and bacteria is what makes us like, yeah, I'm pretty sure.
I'm pretty sure it's Escherichia as well. Yeah, especially because there wasn't much other information on the bacteroides, right? Just the fact that, yeah, sure, it can be in the mouth or whatever, but I mean, you know what else could be in the intestinal tract? Like you said, E. coli. So I think it's probably safe to stick with this answer. Right?
And plus the temperature.
The temperature. Oh, okay. So this guy like has E. coli, as is unknown. But how do you know it's? I thought it's unknown. No, I'm just kidding. So yeah, evidently that looks good. So thanks for telling us that. So there you go. That's how your lab helps. All right, let's look at 23. By the way, I encourage you guys to, so for instance, when you're doing this stuff, when you get to the end of the chapter, I encourage you to look at these questions because they're important. So do that, please. Yeah, all right. And these are most similar to what we're doing anyway, right? So definitely look at these questions, okay? At the end of each chapter, right? Okay. All right, so let's go back. Now we're talking about Enterobacteriaceae. Okay, well, let's look at the families again, right? So we were looking at there we go. It's gram-negative rod, right? Okay, well, let's look E. coli, Enterobacter, Salmonella, Shigella. Let's see. Yeah, they're all there. I remember earlier we were talking about how E. coli and Shigella are genetically related anyway, right? So that's another thing. All right, include many medically relevant microbes or bacteria. We'll check this out. I mean, some cause pneumonia, some cause the plague or pneumatic plague. Some cause urinary tract infections. I would probably argue that these are pretty medically relevant. Right? Okay, because a lot of them cause diseases. Primarily reside in the intestinal tract of mammals. Okay, well, we have E. coli, right? And then check this out. Look at all of these. Normal microbiota of intestinal tract, normal microbiota, normal. They all look like they primarily reside in their intestine of us, right? Are facultative anaerobes. Okay, that one goes back to earlier chapters about instead of obligate, this time we're facultative, right? So that kind of means given the choice, I think we'd rather prefer the no oxygen, right? I believe that's what that's saying. Does that sound right? Okay, good. Yeah, that's why it's facultative, not obligate. Right? Okay, so with that being said, I think these are obligate. It talks about them. Let me see. 286. So sorry, hold on a second. All right, let's go up here a little bit. No. 286. All right, here we go. All right, gram-negative rods, intestine, and it literally means they're in the intestine, right? That's what the enteron mean from entero. And oh, and they also, oh, look at there, coliforms. So maybe that answers our last question, right? They were looking for coliforms in bacteria or in the water. Well, check it out. E. coli falls under being able to make a coliform, right? So there you go. And yeah, look, E. coli and food and water. So I'm just looking for the fact that this can be facultative anaerobe. Well, there isn't much oxygen in our intestinal tract anyway, right? Oh, it does? Oh, there. Yeah. Okay, there you go. So it tells us it is facultative anaerobic. Okay, nice. So all the above. Okay. Genus of gram-negative obligate aerobes. Okay, what does obligate aerobe mean this time? Can't live without it. Gram-negative obligate anaerobe, especially notable in that it is known to parasitize other gram-negative bacteria. Oh, you know this? Oh, it does? What do you think?
I just remember that the one that eats other, like it's that-
Like
that's a virus. Yeah, I know it's A. I don't know why.
No, it's A.
something about
Okay, no, that sounds right. I think
Oh
so.
yeah, I was calling it.
Okay. Even Matthew agrees.
I just
So
remember because the first two letters were weird. So
it does look kind of weird, right?
it stands out.
Yeah.
It's other bacteria.
Right, yeah. So Vibrio is like the other, you know, the sometimes in the waters here we get like the Vibrio vulnificus. Yeah, that's like the flesh-eating one you get. That's not pretty to have. That's really more about your own flesh versus other ones, right? So yeah, I'm sure if you looked up in the book all the other C, D, and E, they wouldn't just eat other ones, right? So, but yeah, I agree with A. Okay. All right, the term algae refers to simple photosynthetic eukaryotes. Okay? And algae differ from other eukaryotic photosynthetic organisms, such as land plants, because, okay, well, let's see here. Organized vascular system, simple reproductive structures. I'm going to say this is probably E, due to the fact that, well, if we think about it, I mean, algae, you know, that's just kind of that collection of stuff, right? It doesn't have a very sophisticated system, right? And also it doesn't have leaves or roots or stems, right? You don't see that. It's algae, right? So I'm sure you guys have all seen it, right? So yeah. Yeah. So this doesn't look very, what the frick? Get out of there. Just kidding. Yeah, so I mean, you have to be careful because I mean, evidently there may be some with some sort of structures, but evidently most of the time they lack all this stuff, right? And yeah, it is true. You can't have unicellular or multicellular. So. Okay. All right. What about this one? Maybe. Okay, what do you think?
I thought out loud. I think it's C. I don't know. It's familiar. I think I've seen this question.
Maybe.
It might not.
I think we should, you know, take a look, right? So okay. Yeah, the Ascomycetes diverse group, and they include penicillium, which, yeah, so why was that significant, that one? What's so significant about what it produces? Heard of the antibiotic penicillin? All right, no. Yeah, it produces penicillin. That was the one where Ian Fleming. No, Ian Fleming. That was the guy who did James Bond. I'm sorry, Alexander Fleming. He did the experiment where he saw there was I think it was contamination, but yeah, basically there was a line of. He had the right, so he had the plate right, and then he was growing bacteria, and he noticed there's just so here's bacteria. There's just some demarcation line, basically, right? Just an area where it wouldn't grow, and that was where the penicillium was, right? It does, right? Yeah, it comes exactly from this one.
There's a movie I
Awesome.
watched before this that really helped.
Oh,
It's called Fantastic Fungi. And it was really
Huh.
interesting.
I have to check that out.
They said that it's actually so amazing, and it talks about penicillin coming from fungi. It's because they have to decompose.
Right.
Plants, they have to also fight off bacteria just like we do, so they actually produce it.
Right. Let's watch it now.
It's on.
Just kidding.
Yeah, it's on Netflix. It's really good.
Okay, I will. Thank you very much. Okay, yeah, let's see. Okay. All right. Not true about fungi. By the way, I think you can say, I think the English thing, they said you could say fungi, fungi. There's several different names that are all correct, by the way. Okay. So, what do you guys think? E. Okay. You guys in your pre-test. Okay, really. I see. Maybe. Yeah. Okay. Right. Okay. Well, chitin, what is that again? I believe it's the matrix, if you will, of cell walls, right? So there's chitin, I think peptidoglycan, and then cellulose, something like that. Excuse me, it's maybe. Okay, it's a polysaccharide, that's right. All right, yeah, structural, that's it, structural ones. Okay, we have cellulose, chitin. So chitin is the structural one, right? And yeah, fungi, see that? Okay, so, well, actually, wait a minute. No, okay, the fungi and bacteria can secrete it, right? So I'm curious, actually. Let's see. Okay, it's a fungi structure. I saw something about reproduction. It said something about, or that was the last one.
It's because there's not numerous.
Oh,
Very, very little.
yeah, I agree with that. Yeah, for instance, maybe the jock itch or the toe fungus, they call it, right? That's probably a couple, right?
Is it in the
Okay, thank you. Oh yeah, I don't really. Okay. Yeah, so I would say these are all true except for, I'm thinking of that chitin is throwing me off. I think I thought they had it. Yeah, okay. So yeah, they have chitin. I'm sure it'll say that in here somewhere.
If you go to fungi and
Oh, they say it.
on the PowerPoint, don't list them.
Okay, probably around here because they start fungi here and then. Oh yeah, okay. Chitin. Okay, fair enough. They do have chitin. Yeah, and those are the one. I mean, you see them on the. Those are the ones that digest the dead stuff, right? That lies on the ground. Okay, and yes, they are. They call them recyclers of carbon and other things because they're the ones who, like the cycle of like carbon. They call it the carbon cycle. They're definitely a part of that. Yeah, you've seen it. So, see that we get how do you think these dead animals are turned into decay products, right? You have the fungi do that, right? Okay, so they can do that. And yeah, the symbiotic relationships, remember the symbiotic stuff? So you can have like parasitism or you can have like commensalism. It's all about working either, so there's parasitism, which is like you have one that benefits and one that's harmed. And then you have, that's parasitism. And then you have the commensalism, which is like the one benefits, the other one is just doesn't matter, it doesn't care. And then you have the harmful one, right? Or excuse me, then you have the symbiotic for both of them, which is the, I think it's called mutual, okay, yeah. So that's where both of them are excited, right? They both get something out of it. Yeah, so with that being said, that looks like, yeah, some of them can team up, the algae and the cyanobacteria and the fungi, and I'm sure they can probably exchange nutrients or something like that. So I wouldn't doubt that can happen.
It's called lichen.
Oh, it's literally called lichen.
It's just what it says. I don't know. I've never heard. I mean, I've heard of the word, but I don't.
Right, okay. I'm liking the idea of that. Okay, yeah, perfect. Lichens. So that's the symbiotic stuff. Okay. Yeah, so this looks all to be true, except for this one. Only a couple diseases. Aflatoxin. Yeah, if you ever take drugs and toxins, you talk about this kind of stuff. It's a cool class. Yeah, even I do know that it is definitely produced by Aspergillus. It's a very potent and very dangerous chemical. Here, I'll show you the structure. Usually they have a number with it, but in general, we'll just look at, yeah, so here's like the B1, right? And does a lot of bad stuff. It's definitely a potent toxic, and it can mess with your, I think, your metabolism or something like that. Definitely, if it does mess with your metabolism, I think. Right. Okay, yeah, liver damage. So yeah, it's just really a toxin. Or maybe I can say, yeah, I think it caused cancer too. Yeah, especially because it messes with your DNA. Yeah, it is a carcinogen. So carcinogens cause cancer. So B is definitely true. It is a toxin. I mean, aflatoxin, toxin. Yeah. It is produced by Aspergillus. That's one of the major, Aspergillus niger, right? That's like the black mold, I think, in some houses, right? In every house in Corpus, if you don't, air it out or whatever. I don't know about the peanuts one. I want to say yeah, right?
I mean, I assume so because if you were right, then yeah.
They can get, that is true. And they can get probably fungal infections from Aspergillus and stuff, right? Okay. I'm going to say yeah, probably E. Okay. Coccidio idomycosis is, okay, let's see. What do you think? Right, yeah. Right, myco means fungal, right? By the way, careful about, remember we said for the mycoplasma, that's not really fungal, that's bacteria. It just so happens that the myco means like, in that context, no plasma or no cell wall. Okay. But yeah, in this case, it's saying there's some disease caused by a fungus. And yeah, it's a fungal disease. Good. So, well, yeah, I guess, yeah, it's a fungal disease. I think it's E. It looks similar, right? Well, we determined, yeah, so the mycosis, that basically means together, the myco means fungal, and the cis kind of means cause of or causes a disease, right? And then, or inflammation too, or that's itis. So. But yeah, it's a fungal disease, and this naming tells us it comes from the coccidia.
Oh, I remember now because it said that when they named them, it would according to where it comes from.
Okay, yeah, so there you go. Wherever the bacteria comes from or infection it causes is what it's named after. So yeah, definitely E. Okay. It wasn't protozoal, right? Because we said it's myco, right? So. Okay, diatoms. Yeah, these are the red tide you see, you know that, the red tide. It can definitely affect you, make your eyes water. So yeah, that, well, it's all of the true except, so one of these is wrong. Algal blooms, that is the red tide thing. Okay, so that is absolutely true. I think, let's see.
I think they have silicone.
You think, oh, silicon or silicones? Oh, no, I don't blame you. Was it it was silicon, right? Oh yeah. Okay, yeah. So diatoms incorporate silicon dioxide into cell walls. And I don't even know what this means.
It's a thing that you use to kill bugs. You can use it in your house.
Okay.
I've actually used it. It's made out of the exoskeletons of insects.
Okay. So for those on the Zoom, this is like a bug killer basically because of the silicon dioxide from the diatoms. All right, and so yeah, looks like that's true, C and D. Looks like the rest, I mean. Okay, that sounds reasonable. Let's see. Oh, here we go. Yeah, algal blooms, that can cause, yeah, you can have poisoning. Although, well, this is talking about zooplankton though, sorry. Does it say that? I don't really see. Oh, here we go. They're heterotrophs in zooplankton that graze upon phytoplankton. Okay, well, both are food. See, that's what they're saying. Both the zooplankton and plankton, phytoplankton are food. I think so. Well, there's. Okay, yeah. So they're saying.
Wait. Oh, that was the first one.
Yeah. So. B. Right. I've heard. I think the holdfast is fungi. Kelp is it?
Familiar. I know it's on the side too.
Yeah, I think holdfast is kelp. Okay. Well, some algae have holdfasts, right? Except diatoms themselves probably don't. You said, right? So, yeah, it's probably B. I think.
Are
Yeah.
they are diatoms?
They are, so, good point, let's see. Well, they say basis, right? So when we think of the food chain, you know, we think of So for instance, it's like inefficient for, let me show you this. All right, so it's actually, when we eat like meat or something, it's actually kind of insufficient because this doesn't really good one, but okay, yeah. So if we eat like the energy from the sun is directly going to like plants and things, and plants are eaten by grasshoppers, and then frogs eat the grasshoppers, right? And so really the exchange of energy is lost every time you go down. So by the time we eat a cow, it's like doesn't even, it's a lot of energy, but it's just not as efficient as eating a salad or something, right? So basically for the primary producers, so yeah, that means like it probably is a producer, see, because it's like producing a food source for other things to eat, right? So I would actually say, yeah, because it looks like the plankton are the ones that eat it, right? So I think this is a primary source of food. But yeah, you're right. Let me just look it up. Okay, the diatomaceous earth is for like filtering. Right? Okay. Excess what?
Where there's excess like
Oh. Oh yeah, good point. Yeah, so it looks like oh, here we go. Okay, no. Yeah, silicon dioxide, sink to the bottom. It doesn't really say anything about a hold fast, right? So I think it actually probably should be B, honestly. Yeah. What do you guys think? B. Oh. So it looks like the plankton.
So plankton are the basis of the food. Or yeah, so plankton.
Oh.
I don't think so.
I think it's saying. That's what I was thinking.
I don't know anything about that.
That's what I was thinking. I didn't see anything about that in here. Well, especially diatoms in general. Sure. First, I want to see if they can, they have a hold fast. Yeah, that's kelp. You were right. So it doesn't, I don't think it does have a hold fast. Oh, and then, yeah, the primary producers. And diatoms are one of the major primary producers in the ocean. So yeah, they produce like food source for all the other plankton and whatnot. So yeah, it actually should be B. Okay. I think we'll do a couple more. Let me see. Oh, there's only 3 left. Sure, let's do it. Okay. Right. Okay. So it looks like those are like a subset. Because this is talking about zooplankton, not phytoplankton. Right. So yeah. Looks like they can be eaten by, and I should look that up. Do eat, what was it? Diatoms, right? Yeah. Okay, yeah, they're a major food source for them. Wow. Okay. So, protozoa.
so it's not like.
Oh, yeah. Yeah, so that is true. Protozoans were talked about. Local.
That's only-
Right. Well, I think they can. Yeah. Okay, yeah. So no you're right, no chloroplasts. So chloroplasts are out. So that is,
And plants can take more.
yeah, exactly. So it is D, definitely. Pretty much just because it says no chloroplasts, right? Okay. Okay, and then as far as the sexual and asexual, I think that's part of the. Oh, there's the diatoms, by the way. Okay, well, yeah. Looks like, so that sexual and asexual form, that's like the alteration of generation or something like that.
And B was on that slide.
Okay,
It was-
okay, perfect. Exactly, thank you. All right,
Oh, wait, no.
what's that?
Well, no.
No, it was. It said
Oh, is it?
they are classified by
Oh,
locomotion. Okay. All right, so the following insect vector. So what do I mean by vector? No physics, but you know. Exactly. It's the thing that transmits it. Right? Okay, so which one is mismatched with the disease it causes, right? Okay. Okay, so it's saying which vector is mismatched with the infectious microbe and the disease it causes, right? C. So I agree that the plague is caused by Yersinia pestis, but it wasn't really a fly going around, right? It was rats, right? Or maybe it was flies that ate the rats, right? Something about that. No. Okay. Alrighty, yeah, so okay, fleas. Yeah, so fleas are not really synonymous with flies, right? Okay, yeah, they do. True. Yeah, and that tsetse fly is like, yeah, it's that louse. Probably some sort of bug, if I'm not mistaken. Yeah, some parasitic insect. Oh, it's lice. Interesting. Lice is the plural of louse. No, I'm just kidding. It does. Yeah. You can still get bubonic plague, like, well, because we take baths regularly. We have sanitation systems. Yeah, see, there's not many in the US, like 1,000 cases, if anything. Yeah, and definitely it's treated pretty fast now. We got a lot of good, yeah, this isn't stuff to mess with. A lot of heavy antibiotics you need. So, yeah, and only this much reported per year per the World Health Organization, which, yep. It's basically gone because, we just have sanitation and all kinds of better cleaning and whatnot. Well, that begs the question, why, how come Ebola probably can't become like a world pandemic? It probably can't. It's kind of related. It's really because it just kills faster than it could even infect, Like once you get it, it's like you got it. I don't think you can spread it without knowing you have it or something like that. Yeah, so yeah, that's kind of how I think that's kind of how the bubonic thing is. Right, yeah, I don't think it's really airborne. Yeah, stuff like that, I think. Okay, well, let's look at the last one. Geosmin is an organic compound with earthy flavor produced by certain eukaryotic algae and responsible for the taste of beetroots and when rain falls. Okay, they're just asking.
You mentioned the rain.
Oh, you mentioned the rain? Okay.
The rain smell.
Yeah, I believe the word for that, I mean, besides this is petrichor or petrichor. Yeah, something like that. Okay. So yeah.
Is it associated with that?
Yeah, let's see.
Oh, yeah.
Yeah. Oh yeah, it's responsible for the earthy. Bro, he got this from Wikipedia, bro. This is like word for word. Or almost. What was that?
Wait, it's caused by streptomyces? What?
No. Produced by,
Oh.
I think so. Yeah, produced by certain eukaryotic algae. Wait, streptomyces.
Is it not?
Yeah. Where'd you hear it was produced by? Okay. Yeah, I don't know about that one.
It was on the-
Right.
I think it was chapter.
Maybe chapter 11.
Wait, no.
This is eukaryotes, I think. Yeah, I don't even see it, so. I didn't, am I spelling it right? Geosmin? Yeah. I didn't find it in the book either, so. Oh, wait a minute. Yeah, he's right. Streptomyces. Oh, yeah. Well, it says streptomyces produce conidia, and then, yeah, they also produce geosmin. I didn't. I'll do that. Yeah, and I'll ask, see if I get the answers. Yeah. Did everyone sign in? There's like 4 people here. Okay, please do. And oh yeah, for you guys on Zoom, before you leave, I have a little sign-in like scan thing for you. So, but yeah, you guys are free to go. And for everyone on Zoom and here, absolutely look at the questions on the end of the chapters. Those are pretty important. Okay. All right. And I guess if you don't have the book, I guess I can send them to you, like a copy or something. Oh, that's right. Yeah, 37 was true, Scott and Cindy. Okay, before you guys leave on the Zoom, give me a minute here. I gotta pull something up. Yeah, I will. Matthew. And yeah, thanks. Yeah, I guess so. Well, thanks for coming anyway, guys. Appreciate it.
Thank you for being an SI.
Yeah, no problem. Yeah, I guess you guys can come to my office hours, but I won't have like actual sessions for you guys.
Thank you.
Okay, no problem. Bye, guys.
Have a good weekend.
You too. All right, I'm still looking for the stuff on Zoom for you guys here. Sorry, there's a little like thing you're supposed to scan. So sorry. Sorry, hold on. Okay. Sorry about this, guys. Just give me one more second. I think it's here. Microsoft Teams. Okay. Okay. Give me a second here. Okay, that's good. Uh, okay. Okay. Yeah, so go ahead and if you have it, get out like the little thing on your phones where you can scan stuff. I'm sure your cameras might have that. If not, it's fine. There's another you can. I'll just link it, I guess, if you can't. But so let me. Okay, so and often this doesn't even work, the scanning thing. So I'm just gonna go ahead and let you go to this real quick. Okay, and I'll go ahead and post it into the chat for you guys. And after that, you're free to leave. I mean, you can leave it. Just fill out the survey, right now, hopefully, please. But you can actually leave right now. You don't have to stay here. Just fill it out. Okay. Any questions before you guys go? Any comments, concerns, questions? No problem. No problem. Okay, guys. I'm gonna go ahead and end it. If you guys don't have any questions. So, oh. Yeah, he didn't give me them, and I feel like that's a teaching thing, how like he wants me to go over them. So, okay, that's good. Yeah, so I'll definitely try to get that out to you guys. Okay. And you can email me. Here's my email if you guys need. Yeah, there's my email. So feel free to send that to me, whatever questions, comments, concerns you have. Ready? Oh, 26. That was. Oh, I gotta pull it up again. Sorry. Give me a second. I gotta stop recording.